This is recording. Okay. Show us not the camera.
Is Vladimir here or David? Balance?
It's just the battery. Oh, there's no battery.
No, this one's supposed to.
Are you showing it?
Where's this one?
I'll start and stop this one. Each speaker. Okay, but it's not. Stop it.
No, keep going first. Until the end of the first speaker. Okay? Sounds good. Stop. It sounds good to you. It's
So the procedure here is that we wait. So we don't advance it. Yeah, we have to realize for the entire thing because we'll rely on this. So just it's not basically on your
Yeah, okay.
Yeah, I got a minute.
Yeah, that's right.
There's space up front. If anybody standing there, space here. What is that? And here and here. Everybody please cluster. Nearest neighbor algorithm. Yeah. Get some more people to say. Okay. This is there.
Okay, welcome back to the second session of Function Cozy. First up, we have Megan Crow, and she's going to talk about predictability of human differential gene expression.
So thank you so much for the introduction. I'm really delighted to be here at ISMB. This is my second time, so it feels like coming home. I'm a postdoc in Jesse Gillis's lab at Cold Spring Harbor, and I'm excited to talk about this work, which was a collaboration with Paul Pavlidis at UBC about, again, the predictability of human differential gene expression. So just to make sure that we're all on the same page, start really basic. What is differential expression, or DE, as I'll probably refer to it interchangeably throughout the talk? It's just a statistical comparison of gene expression between samples from two or more conditions. So here, a really common way of displaying this kind of information is in a plot called a volcano plot. So here each point is a gene. It's plotted with respect to its fold change between the two groups. Here the groups are EGFR positive tumors and normal lung. So if it's higher in the tumor, it's going to be on this side of the plot. If it's higher in normal tissue, it's going to be on this side of the plot. And then the p-value is on the y-axis. And so here I've just highlighted two genes that would be considered, quite good differentially expressed genes. They're very, very different between the two groups. So these are RAGE and hemoglobin. theta globin. And so the way that we typically will analyze these kind of results is to pick some threshold that we consider significant, so either both by fold change and by p-value, and all of the genes that pass these thresholds will be part of a hit list, which we'll then go on and interpret with maybe gene ontology, or looking for gene family enrichment, or maybe for KIG pathways, whatever your favorite tool is, or maybe whatever you have on your server already. And so this is kind of the typical way of looking at things, but of course today we're at function sig, and so another way of looking at this is to actually tip it on its head. And instead of thinking that we need to interpret this with additional databases, maybe we can think about these as annotations directly. And what would that tell us? So now we have this list of genes. We can instead think about them as maybe as lung cancer genes, or if we wanted to sort of lump everything together. And now we can start representing it the way that we think about feature sets more broadly, or annotation sets more broadly, where we have a vector of genes for this data set. If a box represents a single gene, and if it's red, that shows that it's DE. And so thinking about it this way sort of opens up new ways of thinking about this kind of data, and maybe CAFA-like starting to think about predictions. Okay, so our question here was, how well can we predict differential gene expression? And if we can predict DE, what drives performance? What would that tell us about the expression data, sort of both from a biological and a technical perspective? And so the general approach that we're going to take is very simple. So this is the analogy we like to think of is that of a psychic, right? So the way that a psychic makes predictions is just to make really, really generic predictions. They know that in general the population will have a propensity toward certain maybe fears or ambitions, and uses that to make something that sounds quite plausible. So we're going to try to do this, except not for personal characteristics, but instead for DE hit lists. And we're going to do this for all available human microarray data sets, and trying to predict what genes are DE without knowing anything specific about the goals of the individual experiments. Okay, so the next two slides are just my data and the methods, and then we'll get into the results. So the data that we're using, and the reason that it was such a great opportunity to work with Paul, is that we have all of the data that they've been curating as part of the GEMMA database over almost a decade now. So they've been collating publicly available human and actually expression data from other species as well, but we're focusing on human in this study. They have consistently reprocessed this data. It's been manually curated, so there are a lot of great tags. It's annotated for different ontologies, for example, and then there's also batch correction when it's necessary. Then differential expression is calculated with very standard methods, linear regression, and then, of course, there's corrections for multiple hypothesis testing. So starting with this big compendium, we needed to think of ways to filter it in order to kind of avoid biasing ourselves. And so we had initially 2,500 different data sets across 10,000 different conditions. And so the first filter that we wanted to implement was that of trying to avoid gene representation biases. So we sought out just a platform that had the most data sets and the most genes represented, which is GPL 570. That limited us now to 883 data sets with 3,000 conditions. But you can imagine, so when I'm talking about conditions here, I mean that one individual experiment might have looked at, let's say, 4 different time points. But if they all compare back to time point 0, those conditions are not going to be independent. And so to maintain sort of strict independence within our compendium, we only wanted to look at one condition at a time for each data set. And so we chose the condition that had the most samples as a way of trying to limit this kind of an issue. And then finally, of course, we're interested in differential expression, so the data sets had to have at least one differentially expressed gene. That left us, again, we're implementing the same kind of thresholds that I discussed at the beginning. And then that left us with 635 total data sets, and which was about 27,000 samples collected over many, many years on this one platform. And if you want any more details, I should point out that this is published, and you can feel free to look it up and ask lots of questions. Now getting back to what the method was, this will be familiar to many of you who might be familiar with Jesse and Paul's earlier work on multifunctionality. The sense of it is that we're counting up the number of times that a gene is differentially expressed within the compendium, except that we have a trick that we want to weight it by the number of genes within the data set. So you can imagine that you would want to weight differently, and because we know that there are different numbers of DE genes within each of these hit lists, we need to consider them differently. And so that's what this, this formula shows at the bottom here. So for each gene, we're going to give it a score for one data set, K, that's 1 over the number of positives times the number of negatives, so positive, differentially expressed genes, not differentially expressed genes, summed over all K, leaving out one data set at a time that we're going to predict. Okay, so we have these scores, we rank them, and then we use that as our predictor. And the performance is going to be expressed throughout as the AUROC. You can also think of this as the proportion of correctly identified genes. Okay, so now for the rest of the talk, I'm just going to be focusing on the results. Excuse me for 1 second. Okay, so the basic characterization, the basic result, is that DE gene lists overlap. We see, so here the histogram shows the x-axis. This is the number of times a gene is DE within the compendium. This is the frequency for all genes. Most genes are differentially expressed at least about 10 times out of our 635 data sets. But you can also see that there's this sort of long rightward tail. And so if you wanted to be act like a psychic today at the poster session, you might go around and ask people if they found CXCL8 in their DE hit list. But the fact that there is this sort of long tail isn't itself surprising, but what that does suggest is that there might be this kind of generic signature that we can predict. And so now when we sort of transform this into our AUROC metric, where we're looking for the ability to predict these DE hit lists in all of our compendium, this is what our performance histogram looks like. So here, the x-axis is the AUROC, the y-axis is the frequency in terms of data sets now, and you can see that we're very far from, on average, very far from just sort of randomly guessing what genes are going to be differentially expressed. We get an ROC of about 0.8, so we can predict about 80% of the differentially expressed genes. Okay, so coming back to our questions, how well can we predict differential gene expression? Turns out, pretty well. And so now that we've shown this, what drives our performance? What does that tell us about expression data? And so for now, I'm just going to show you the kind of biological story when we look at the top-ranked genes. And so we did a lot of different controls to try to make sure that we weren't overly biased toward any particular known biases of microarrays like GC content or length, but none of those things seem to have as much performance as the prior. And so when we looked at what the top genes were, they actually made a ton of sense. They were things that we would expect to see a lot of the time. So here are the top-ranked genes. We've looked for their co-expression in an aggregate network with totally independent data from RNA-seq, and then we've clustered them by their co-expression links. And when we look at the enrichment for the clusters that we see, first of all, we see they're not super distinct from one another except for this Y chromosome gene, so there is some co-expression. We do expect to see co-expression of these genes with one another, but there are some distinct processes going on. So we see some maybe developmental or proliferation associated processes over here. We see stress responses, so immediate early genes are among those, and then also inflammatory and interferon responses. So these are among the most commonly differentially expressed gene processes. And the way that this feeds back into our analysis, when we go back and look at our data sets again, and here I'm showing each column here is one of our DE hit lists. It's colored by which NIH agency funded it. And so you can see that it actually doesn't cluster at all by the NIH agency, but instead there are these sort of complex patterns where individual studies are enriched. So the boxes here, if it's black, that means that it's in the top 1% of all enriched functions for that data set. We can see that that there are these sort of complex patterns of having enrichment for maybe one or many of these different modules, suggesting that it is sort of this combination of effects that allows the prior to have such high performance. Okay, so what can we do with this information? So I mentioned that this is just simply a ranked list of genes. This is the top 20 list of genes. If you are like looking at gene names, you can see that these are sort of the Y chromosome ones. Again, EGR 1 and 3, very sort of stress responsive genes, FOS as well, and then CXCL8, IFI 27, again, interferon and inflammation associated genes. So this ranked list we can use now to interpret hit lists from individual studies, just like we would do with any other ranked list we might have in bioinformatics, right? So we might, just like we would use, let's say, percent conservation between two species, or we might use the mutability of a gene within the general population with something like the probability of loss of function interaction from exact. We can use this generic likelihood of DE to sort of sift out differences in our gene lists. So I'm going to show two examples of this, two applications. And so the first is a set of housekeeping genes. These were identified in a study where they were trying to look for the stability of genes in single-cell RNA sequencing data. The y-axis here, so each dot is a gene. The y-axis here is the rank of that gene within the prior. So if it's toward the bottom, that means it's less frequently DE, and if it's toward the top, it's more often DE. And so nicely, we see that most of these genes that were considered to be possible housekeepers that we might want to use to normalize our data are actually very infrequently differentially expressed, which is great. But of course, we would be worried if we wanted to normalize against these ones at the top, which are very frequently differentially expressed. You can imagine if you use those to normalize your data, you might mask some of the condition-specific variation because they are very, very commonly differentially expressed. And these include ApoD, I believe, and RPS24, something that sounds like perfectly innocuous ribosomal protein and instead might be, in fact, worrisome for your normalization. Okay, and now coming back to this disease-associated kind of analysis. Here we have done a meta-analysis of lung adenocarcinoma, same example that I gave on the very first slide. Here are the same two genes. We see across the x-axis, this is the recurrence of genes in different data sets. These are very, very recurrent genes, so again, quite good genes maybe for eventual follow-up. But then what we see is there's actually a bit of a split between their likelihood of just generically being differentially expressed, where hemoglobin comes up in many, many other conditions, whereas RAGE might be considered more specific. So this is just a way that you can then go forward and interpret your results and maybe decide whether you wanted to go a more specific route or a more generic route. Okay, so in summary, we found that many genes overlap between DE studies, which makes it easy to predict a large fraction of genes using a single ranked list. We think that the DE prior is high performing because it captures a combination of very common biological effects. And finally, we find that the DE prior provides context for hit lists, which let you say whether a gene is more or less expected to arise or more or less specific to the phenotype. And finally, I need to acknowledge my lab, my supervisor, Jesse Gillis, Paul, and Nathaniel. Please visit Nathaniel's poster, C17, which is an extension of this work. If you'd like to read more and download the prior and try it out for yourself, you can. This is the paper. And of course, I need to thank you all for your attention and the funders for the chance to be here today. Thank you.
Okay, thank you. We have time for some questions.
Questions? Any? Yeah, this work. Anyone? All right, I'll right here.
Great work. The XIST, like the X activation thing right at the top was
Yes.
pretty striking, so it's definitely working. So the 17% of genes that you can't predict, like, do you think they're more interesting, or have you done, have you looked into what those genes are in general?
Right, that's a great question. It's not something that we've focused on, actually, so that would be a great next step, I think. I think in general, our feeling is that we're just sort of we're like on the edge of being powered to do this analysis, so we were very happy with the degree to which we could do it at all. But yeah, but I think that would be a great thing to look at.
Any other questions? One over there. Raise your hands high. So, yeah.
Hi, I have a very small question. Why did you take the log fold greater than 2? Because it's quite a high threshold. Because usually you either take it 1 or 0.5. And the next question is that why didn't you take it like less than -2 to find the down-regulated genes?
Right. Okay. Yeah. So the question was, why did we pick such a strict fold change threshold, and did we look at negatively and positively regulated genes together or separately? So to answer the first question, we did try a couple of different thresholds. Initially, we had chosen log greater than 2 because we had actually included some RNA-seq, and that's the CQC threshold suggested. In the end, we decided there were this project spun out in lots of different ways, but it's sort of a legacy issue, I would say. But in the end, we found that when we chose a different threshold, that we actually had very, very similar results. So the prior itself is very highly correlated, whether you pick a fold change of 1 or a fold change of 2. Fold change of 2, I think, is really getting at the question that we want to answer, which are what are the really high, strong effect size genes that are always going to come up. Yeah. For the second question, between positively and negatively regulated genes, we would have loved to do that, but even though the manual curation in GEMMA is wonderful, it's not always consistent. And so you could imagine for the Y and X chromosome, sometimes females are the control and males are the condition, and sometimes it's the other way around. So positive and negative has no meaning basically in this data. Yeah, it can go both ways.
Okay, thank you very much.
Thank you.